so much, Salar, for the nice introduction. And I would like to thank to Drs. Atharinam and Professor Zafar Bashir for having me and inviting me for this talk and introduce myself and show some of the work I've been doing in the last few years since I'm here in the AQU CRM. And also discussing some of the latest technology, which is actually growing very fast in terms of understanding the brain diseases, especially in vitro model system. So here's the talk. The title I am going to present is Organoids as a Model of Human Brain Development, Neurological Disorders, and their impact on the clinical application. So this is a brief outlines of my talk, which I'm going to introduce some of the background and about the organoids and some history, historical journey of the iPSCs derived organoid and how to generate these organoid in the dish, and some of the disease modeling. And finally, some application we can utilize in any context of the neurological diseases. So we all know the brain, human brain is very complex, and it still is the mystery how to discover it in the context of the in vitro system. So human brain constitutes the cells across the several functions and is interconnected with structures from neuroectoderm structure and substantial distinct compared to the other species. So human brain is responsible for perception, cognition, and some major function which the other species we cannot study through them. So how to understand the human brain, which has a complex diseases, as you know. The most of the complex diseases we are facing in current time with the neurodevelopmental disorders, neurological disorders, and some psychiatric disorder. And the last 2 years, we are facing this viral infection, such as COVID, coronavirus. And then some people are affecting with the brain tumors. All these complex diseases, but we don't have a much model system to understand and cure these diseases in a short period of time with the less economical condition. So how to study human brain in a dish? That's the question we all have in every day when we do the research in the lab. So there's a big difference between the human and the other species because of the complexity, due to the different cell type and due to the size of the brain of human and the number of neurons and number of connections and number of different functions which a human brain do in the daily basis compared to the other species. So we need to understand all this complexity in a dish to utilize the special model system to answer all these questions which we have. So here's an example, two different species, for example, the rat and the human. As you see, they are going through the same developmental stage, but there's a huge difference between the mouse or rodent's brain compared to the human brain because of this morphology composed of the cortical gray matter, which is the huge, and all these folded region, which mouse brain does not have it. So we cannot utilize some of the diseases such as microcephaly or lissencephaly, which are also early developmental issues. And the species-specific different cell types, for example, the radioglial cells or intermediate progenitor cells and some ventricle zones. I'm going to give you some brief description of the cortical region, which I am very much interested to understand how the cortical region is actually developed. So here you see the to comparison between the mice cortex region and the human. On the left side, you see the rodent brain, which is started from the ventricle zone, and all these cells which born or stem cells born from this region, and they divide, migrate, and they reach to the cortical plate, where all these newly born neurons resides, and they become a mature, and they're forming these different layers. As we know, the cortical regions comprise of the 6 different layers. The difference between the human brain comes when it comes to the 2 different region, which is like outer subventricle zone, which divided into the inner subventricle zone and outer subventricle zone, where the most of the intermediate progenitor cells is actually after they come out from the ventricle region, and they reside in this region before they migrate and differentiate into this multiple cell types, such as intermediate neuron or mature neuron, astrocyte or oligodendrocyte. So this huge difference between these 2 species. So some of the diseases, for example, the microcephaly, we cannot study using the mouse brain. So it's not the good model system. As you see, this microcephaly happened because of this NDE1 gene mutated in the human when the child born with this mutation. So here's the left side. You see that this is the normal brain MRI scan, and whereas this bottom one is showing the 4.5 years old, and it's microcephaly, means like the small brain size, which does not grow in the normal pace. When the scientists decided to utilize the mouse model system, and they knock out this gene, and they did not find any difference between the wild type and the knockout mouse brain. They looked very similar. So it turned out we cannot utilize this model system to understand some of the diseases. In the everyday or in the current situation, most of the labs are actually utilizing the model system in the life sciences, such as the 2D culture or the spheroid. You can just take the samples from the adult tissues or primary tissues or primary culture and get the cell aggregation. And we also utilize the patient-derived xenograft model. So these models still utilized in the many labs. But there is a complexity between the 2D culture and the 3D cultures. As you know, the 2D culture is a lack of complexity, they're easy to use, and it's also cost effective. When it's come to the 3D culture, it has a increased biological relevance, which is very similar to the in vivo system. And they have more cell-cell interaction and the more complex, but it costs a little bit high. But it gives you the closely or recapitulating the organ system, which we try to understand. So different techniques represent the trade-off between homogeneity and complexity. It depends on what project requirements. So we also have some limitations. So utilizing the different sampling, for example, we do not have any access of the human brain sample. And some of the mouse model, we cannot utilize those mouse model because some specific traits we cannot study on the human on the mouse model, such as if you are studying in the neuropsychiatric disorders such as autism, and the autistic children has a communication problem. So how we can utilize this mouse model, because the mouse does not communicate or they cannot talk. And the same thing goes with the, we do not have any brain bank. And if you utilize those postmortem tissues, first of all, they have a lack of information because they preserved often does not represent the stages of the diseases when you are looking for. Which it does not actually give the information, some of the neurological, because it's the end stage of the diseases. So these limitations actually does not allow us to go further to utilize them. So what's the solution? And still we have this question, how to study human brain in the dish? So the embryonic stem cells actually comes, this is the cartoon. I'm just showing you some of the people, they may be interested to understand how the embryonic stem cells is actually come from, and they are the pluripotent stem cells. You see, this is the egg. They started developing after the fertilization. So this is a 5 days stage in the human, and the stage comes to the blastocyst, and this is the whole blastocyst where the inner cell mass consists of almost 150 to 200 cells, which is called inner cell mass, which usually comprises these pluripotent stem cells, and you cannot barely visible when you look into the microscope. So how these pluripotent stem cells is actually utilized for the study in the modeling, because these pluripotent stem cells, they have ability to self-renew and unlimited, and they have ability to differentiate into multiple tissue type. As you see here in this cartoon, so these blastocysts or inner cell mass or pluripotent stem cells, you can just take out these cells and grow in the dish, and then differentiate into the multiple lineages. It depends what you are interested in. And then utilizing these pluripotent stem cells to grow the organoid in the dish. But again, we have challenges. So what are the challenges? These challenges are the ethical concerns, because we cannot utilize human embryonic tissue. And we cannot get it because it's a human early stage of the human tissue, which is a day 5. And it's also risk of immune rejection or genetic compatibility. So what are the solutions? The solution is actually come out when the Shinaya Yamanaka in 2006, he got this Nobel Prize in 2012. He's the revolutionizing study after generating the induced pluripotent stem cells. I'm just gonna go through quickly how we get the induced pluripotent stem cells in the dish. So we can get the biopsy or skin cells from the patient and the healthy individual. And then these skin biopsies, you can convert or grow them into the fibroblasts or somatic cells. And these cells are reprogrammed by the multiple transcription factor we call the Yamanaka factors. And those Yamanaka factors actually convert to these fibroblasts into the pluripotent stem cells. We just learned from the embryonic stem cells in the previous slide. And these cells has exactly the same capacity to directly differentiate into the multiple lineages, as you see here. So you can get a different tissue type from these pluripotent stem cells. And then we can use these pluripotent stem cells into the organoid in a dish. And these organoids can be used for the drug screening or drug-based treatment or cell-based treatment. So now we have this in vitro model system. This model system allows us to understand or discovery of this stem cell, improve the organoid technology and eliminate all the limitation for the in vitro model system in the laboratory environment. So here I'm just gonna go to show you what are the organoids basically. So organoids are the 3D stem cell or tissue derived structure. Most people commonly call them mini organ in a dish. They mimic the in vivo architecture of tissue of origin structurally, functionally, and genetically. And they have a powerful means of ex vivo modeling, as an in vitro model system of stem cell research and the developmental biology. And they can you can derive them from the embryonic stem cells, induced pluripotent stem cells, and some of the adult stem cells. And as you know, they are the self-renewal and self-organization. So there is a multiple advantage of utilizing these organoid model system, because they are very close to the physiological model, and they have a genomic stability, and you can study any human diseases in vitro, and they can utilize for the regenerative medicine. So the timeline of the development of the generation of organoid technologies actually started in early 1900, when the first time the self-organization of sponge cells, they showed the ability to form the aggregation. And then another group showed this, these aggregation is actually required some the extracellular matrix system. So from the 1900 to the now, this technology has grown really, really fast, and still they are growing, and we have multiple tissue type of organoid model system, and we can utilize the different disease modeling in the dish. And multiple group actually got on this Nobel Prize in the past also. So I'm going to talk about the different diseases, as I mentioned earlier. So some of the neurodevelopmental disorders, you can study the viral diseases caused by the different viruses, such as the Zika virus. And a few years ago, there was a big group in Brazil. They studied first time utilizing the Zika virus effect on the organoid model system. And the SARS-CoV-2, since 2020, the lots of people actually have done utilizing the organoid model system to understand the effect of this, the coronavirus on the human brain. And multiple groups are studying the neurodegenerative disorders. And the last is the brain tumors, which I'm also very interested in the future to study utilizing the organoid model system. So the cortical organoids, as I said earlier, is one of the region. The cortex is the which comprises of the two lobes, and cortical region, which consists of multiple layers. Organoid in the dish, now you see almost their size is a few millimeter in size. This is the early stage when you see all these rosette formation. This is the sign of they are in the right direction. So human cortical organoid, they recapitulate the early event of brain formation, and they are used as a model of the multiple cell types, and their architecture relevant to the carrying out the function, and they are useful to further studying the homeostasis, regeneration, and the diseases. The imaging of organoid is allow you to visualize and understand their self-organization and symmetry of breaking the growth, differentiation, and the multiple cell types. So once you have these organoids, and this is a picture of representation images of the cryo sections. One of the big 5 stage. How you gonna confirm these? This is the cortical region, or this is the cortical organoid region. So I utilize one of the markers called the FOXG1, and then confirm these cortical sections by the confocal images, which showed the high expression of FOXG1 together with the SOX2, which is the early neural progenitor markers, which confirm our organoids is actually inducing towards the right direction. So this is just a cartoon. I'm just gonna be already learned briefly in the previous slides how the cortical region is actually expand and comprises of different layers formation. So stereotypical organization, and how these neuroprogenitors is actually behaving when you study in the dish. So there's the multiple markers. They've been utilized for the identify these markers, and these markers is already established markers to compare these sections in vitro system. And you also label these marker if you are interested in a different cell type or different either in neuroprogenitors to the mature neuronal cell types or astrocyte or oligodendrocyte. So what I did first, I just wanted to see the time point experiment, and these organoids is actually recapitulating the architecture of the early brain development, which I found, yes, they did. Actually, SALB2 is one of the marker, which is the mature neuron markers, and it does not have any expression pattern in the week 5, which is the very beginning of the brain development. But by the time of the week 10, their expression is exploded, and we lost all these rosette formation, which is recapitulating the early cortical or early ventricle zone region. And some of the markers were utilized for confirmation of the neuronal lineages, such as the CTIP2, which is the deep layer neuronal marker, and UN, which is a very specific neuronal marker. Again, I also utilized the radial glial marker, which is called the HOPX. And this is also born in the ventricle zone. And these are the radial glial marker is actually the one very specific to the human development. It play a very important role in differentiating in multiple cell types. Again, it does not express in the early time point, but it expresses in the later time point because it's one of the marker which started coming out after the ventricle zone. All these progenitor cells is actually born, migrate, and reside in the subventricle zone, and then further differentiate into the other cell types, such as the TBR2, which is also upper layer marker, a neuronal marker. So I just found a very similar pattern in the dish in the organoid system. The maturation of the cortical region or cortical organoids, it comes to the stage of the week 10, which this 2:1, which is it's a neuronal maturation marker, and you see by the time of the week 14, which is the very late of this organoid which getting mature, and we lost this ventricle zone, and all these cells is actually started to differentiate into the neuronal lineage. And then I also applied the S100 beta. It's one of the astrocyte marker, which highly expressed in the both started coming to the week 10, and it's exploded by the week 16. And it turned out all these systems giving me the same spatio-temporal or architecture of the brain development, which we are interested to understand the early brain development. So now I'm going to introduce one of the things which is the hot topic of the last couple of years since the corona came in our lives, and it causes so much problem to many people who got affected. So, and lots of neuroscientists started looking the coronavirus pathology, or how this coronavirus is actually infecting the human brain, how it is crossing the blood-brain barrier. So here you see the olfactory nerve. Once the coronavirus is attacked, the human goes to the blood-brain barrier system. Is some of the leakage system, and is embedded to the brain, because the blood-brain system comprises first line of the microglia and astrocyte. So astrocyte is the one of the cell type is actually at the front line of the blood-brain barrier. And we found this astrocyte, they infected a lot when this coronavirus was infected to the organoid system. And this paper was published in last year, actually, and utilized this viral infection to human stem cell derived cortical organoid culture. and the different time points, this we actually looked at the effect of the coronavirus, and we found the astrocyte is one of the cell type in the brain which is infected by the coronavirus, and it compromised the blood immune system through the leaking of the blood-brain barriers. So finally, I'm going to talk a little bit about the application of the organoid technology. So considering the advantage of the organoid, which offer the applications of multiple fields, as you know, like we are interested to understand the host microbe interaction and the modeling the cancer metastasis. And these organoid model also utilized for the gene therapy by you can either delete it or knock in the utilizing CRISPR gene technology. And importantly, in the field of drug screening, the organoid model is very close to the in vivo model system. So in coming years, the potential of organoid models for the multiple neurological diseases, which you see in this figure, you can either utilize for the neurodegenerative diseases or neurodevelopmental disorders or the brain cancers. So here I'm going to just go through the little bit about the preclinical application, the biobanking of the glioblastoma organoid system. So the glioblastoma is one of the cancer which infect the brain really fast. So how are we gonna study the glioblastoma in the dish? So you can utilize the glioblastoma organoid, and you can genetically engineer, and then you can do some of the modeling, either studying the survival and proliferation, drug screening and invasion assay, and the patient-derived xenograft mouse model. And importantly, these sample can be used for the biobanking. For this group in Stanford, they work another level of 3D model system, which is called the assembloid. So because the brain has a multiple region, and each region has a very specific function, and they also communicate each other. For example, in this paper, they've studied the two different regions, which is the here you see this cartoon. This is a cortical region, and this cortical region is also connected with the striatum. So how are you going to study the cortex and the striatum, how they communicate, how the cells migrate from the striatum to cortex? So you need to establish, or you need to develop the model system to study the communication. So what they did, they utilized induced human pluripotent stem cells and generated two different kind of organoid, and they grow them together, and they form the symboloid in one well. After doing this, so these two different type of these organoids were labeled with a specific labeling. For example, here's the one M cherry and the one YFP. And after certain time period, you see the projection of the human cortical organoids, that these cells is actually communicating towards to the other side of the organoids, not the vice versa. So it's mean the communication is actually starting going towards the cortex, not from the cortex to the striatum. So this is a very neat model system to understand the communication between these neuronal cells, and they are and the striatal projection in the human forebrain or human brain into different stages of the development. So recently, this group also working on the multiple assembloid or triple organoid system. So as one of the example is a neuromuscular junction, and this neuromuscular junction is one of the system is the very is a peripheral synaptic connection between the motor neurons and the skeletal muscle fibers, and that control the movement. So how do you study the motor neurons and connected to the cortical region, and the same time how they project each other, the communication. So here you see the you generate all three different type of organoids and utilizing this initial induced pluripotent stem cells and the skeletal muscles tissues utilizing, and then generating this organoid model, and then combining all these three organoids they generated from the different areas of the body. And you see the beauty of these organoid systems, which is a triple organoid, and they're all connected to each other and they're communicating. So it's efferent and efferent communication, which is comes through your motor neurons and go to the muscles. And this is the one of the future of the organoid modeling in the dish, and we are hoping this field is growing faster enough, so then we can understand the brain much better and the brain function, and also some neurological diseases in the dish, so then we can identify some of the treatment. So I'm going to summarize my talk. As I said, these are the organoids. We are interested in neurological disorders and the neurodevelopmental disorder. They come from this pluripotent stem cells, and you see all these directions which I have talked about. Either you can utilize these organoid model system to gene editing and the disease modeling, and also assembloid functioning. You can understand the different function of the cells or neurons in the communication, and also some transcriptive analysis and epigenetic analysis of utilizing like a single cell RNA-seq. So, and doing that, you understand or you get some more information about the different cell types and different signaling pathway, gene expression, and some of the genetic profile, doing it with the transcriptome analysis. And finally, this is I'm going to summarize further all these I'm not going to talk about, which I have already said, the disease modeling and drug screening, identification of the molecular mechanism, and the host, viral infection, and regenerative medicine. This is the organoid can do in the in vitro model system. So I'm going to acknowledge a number of people. The first, I'm going to say thank you so much for my departmental interim committee, which they've been very supportive and giving me so much support doing my work here at the CRM, and my co-faculties and the staff members at the CRM and the core staff. They've been very helpful during my work, and the admin staff and some of my collaborators around the globe. Thank you